This diagram illustrates the central paradox of cell biology, fitting 2 meters of DNA into a microscopic nucleus without it becoming an impossibly tangled mess. To achieve this, the cell must organize its genetic material through several precise levels of physical condensation. As we split this chromosome apart, you can see it is not made of naked DNA alone. Instead, the cell utilizes specialized structural proteins to act as the framework for the entire molecule. This is the baseline chemical composition required for the WBCHSE exam. A chromosome consists of DNA combined with 2 distinct classes of proteins, basic histone proteins and a diverse group of non-histone chromosomal proteins. Understanding how these proteins interact to fold and coil the DNA thread is the first step in mapping the structural hierarchy of the genome. This diagram shows the nucleosome model, where the first level of packing is driven by simple electrostatic attraction. The DNA phosphate backbone carries a strong negative charge, which is magnetically attracted to the positively charged basic amino acids, specifically lysine and arginine, found in histone proteins. The core of this unit is a histone octamer, composed of exactly 2 copies each of H2A, H2B, H3, and H4. A segment of DNA approximately 146 base pairs long wraps around this core 1.65 times to form the nucleosome core particle. Notice the separate protein snapping into place where the DNA enters and exits the core. This is histone H1. It acts as a structural clip to secure the linker DNA. Remember, H1 is essential for stabilization, but it is not part of the 8-protein octamer core. This repeating beads on a string arrangement converts a long, fragile DNA molecule into the fundamental structural unit of chromatin. As the cell condenses further, these beads on a string coil together a structure known as the 30-nanometer chromatin fiber. Chromatin exists in 2 distinct states within the nucleus. On the left is euchromatin, where the coils are loosely spaced and lightly stained, making the DNA transcriptionally active. On the right is heterochromatin, which is so densely packed that it appears dark under a microscope and remains transcriptionally inactive. By physically tightening or loosening these fibers, the cell regulates which genes are accessible for expression and which are silenced. At its most condensed state, we reach the metaphase chromosome. Looking at this X shape, it is important to distinguish the vertical halves, which are called sister chromatids. While chromatin refers to the DNA protein material, a chromatid is 1/2 of the fully duplicated structure. The point where these chromatids are joined is the centromere, or primary constriction. Attached to the centromere are the kinetochores, protein discs that serve as the attachment site for spindle fibers during cell division. The ends of the chromosome are capped by protective sequences called telomeres. Additionally, some chromosomes feature a secondary constriction that leaves a small bulb of material at the tip, known as the chromosomal satellite. Before we classify these shapes, let's test your recall of the nucleosome's anatomy. Look at this diagram and identify the core octamer, the wrapped DNA, and the external H1 protein. This diagram categorizes 4 chromosome types by centromere position. Metacentric has a central centromere, making a V shape. Submetacentric is slightly off-center, forming an L shape. Acrocentric is near the end, creating a J shape. Telocentric is at the tip, appearing as an I shape. To summarize, we can trace the structural hierarchy from the fully condensed metaphase chromosome back through the 30-nanometer chromatin fiber, the repeating nucleosome units, and finally to the original DNA double helix. Mastering this progression from the initial electrostatic winding of DNA to the final organization of metaphase chromatids provides the structural foundation for every chromosome question on the WBCHSE exam.